Professor Carrington.
Thank you. First of all, let me thank the organizers for inviting me to participate today. And it's a pleasure to be here, and I look forward to the discussion afterwards. So I'm going to just speak a bit about both dengue and chikungunya. In chikungunya, in particular, I'm going to focus on the patterns of spread globally and within our region, and what we have been doing to try and understand the factors that underlie the rates and directions in which dengue moves when it once has been introduced to a region. So we've got some background on dengue already, so I won't dwell on this. It's the most rapidly spreading mosquito-borne viral disease, and it's one of the most important emerging diseases of the 21st century.
Sorry, Dr. Carrington. Sorry, Dr. Carrington, could I ask you to pause for a moment, please? And we're just asking them to resend the presentation.
Okay, we're getting it.
Okay, we're getting it now. Okay, thanks. Please go ahead.
Yes, so dengue itself is caused by the dengue virus. It's a virus that belongs to the genus Phylogenetic, and this family contains viruses with RNA genomes. Now, you heard before that there were 4 serotypes. That refers to these 4 serotypes that are associated with epidemic disease and the 4 serotypes that are found in the region. However, recently there was a report of a 5th serotype identified in Malaysia, but this serotype appears to be symbiotic. The full report on this serotype is not out yet, so we look forward to it with bated breath. Now, you have these 5 serotypes, and within each serotype you have different genotypes. So you can identify different strains within the different serotypes. We're familiar with the outcomes of dengue virus infection. It was mentioned briefly before. You can have a relatively, 80% of cases are asymptomatic, so you can get infection and have absolutely no symptoms at all. And this is a very important consideration, because it basically presents a reservoir, an undetected reservoir of infection that can be transmitted from one person to another. So it's a pool of uninfected individuals that are very important in the epidemiology of the disease. It's a pool of asymptomatic individuals, I should say. So if symptoms do present, you can get a range of a clinical spectrum of presentations. You can have dengue, classical dengue, with or without warning signs, or you can have a very severe form, formerly known as dengue, hemorrhagic fever dengue, shock syndrome, but now known as severe dengue. Now, upon infection with any one of the serotypes, a person will be immune to that serotype for the rest of their life, and you'll have limited but short-term immunity to other serotypes. However, infection, that doesn't last, and infection with other serotypes is possible, and as we are familiar with, there's an increased risk of severe manifestations with secondary infections. Chikungunya now, which, as we heard, is on our doorstep, is a re-emerging mosquito-borne virus in the old world and recently emerged in the new world. It is widely distributed, originally centered around Africa and Asia, and moving outwards and now in the Caribbean. It has many similarities with dengue in that it uses the same vector, dengue, and the disease manifests similarly in terms of it being a febrile, starting off as an undifferentiated febrile illness, and there's a lot of overlap between the symptomology, which means that's often misdiagnosed. It's a totally different virus, however, it belongs to the genus alpha virus, also RNA viruses, and there are 3 genotypes that exist. As far as we know, there's 1 serotype, but 3 genotypes within that serotype. The Western African, the East Central, South Africa, and Asian genotypes. So chikungunya fever is the typical symptoms are high fever, joint pain, joint swelling, rash, headache, muscle pain, etc. It's rarely fatal, unlike dengue, which can be life-threatening. And the acute symptoms typically resolve in a short period of time, but there are cases of people who experience persistent joint pain for months or even years after. Now, both dengue and chikungunya are transmitted in an urban epidemic cycle by both with Aedes aegypti and Aedes albopictus as the main vectors. So the urban epidemic cycle is what occurs in the Caribbean, but there is also a sylvatic cycle for both viruses, where the virus alternates between infecting forest-dwelling mosquitoes and mammalian species, of most primates are the main reservoir. Now, in the case of chikungunya, this sylvatic cycle occurs in Africa, and it is where the virus is maintained during inter-epidemic periods. So you have a sort of enzootic sylvatic cycle maintaining the virus between outbreaks. In the case of dengue, the sylvatic cycle exists in both Southeast Asia and West Africa, and the currently circulating dengue 1 to 4 viruses that we associated with epidemic disease arose as a result of successful spillover events that occurred hundreds of years ago. And dengue made that successful jump from a sylvatic transmission cycle into human populations 4 times, and each jump resulted in the emergence of one of those serotypes. And we now have evidence that there's a 5th one occurring as well. So this figure that I'm showing on the left here just shows the relationship between the sylvatic viruses, the genetic relationship between the sylvatic viruses and the epidemic viruses in circulation. And you'll see that each one of the serotypes is closely related to a sylvatic strain. Now, having jumped over into humans hundreds and hundreds of years ago, dengue was mainly a virus that you would see in Asia. It is thought to have originated in Africa, moved over to Asia, and then from Asia you got that expansion, the sort of global expansion of the virus. And since the 1950s, there's been a dramatic increase in the incidence of dengue disease, and in the number of countries reporting the number of serotypes circulated in different regions. Within our region, we got a bit of a history of the circulation of dengue viruses in our region, and I'm showing this here. What I want to highlight here is the fact that our history of dengue in the Americas is basically a history of repeated introductions of viruses from Asia. So we have had dengue within the Americas for hundreds of years, but we only had dengue virus serotype 2, and that was thought to have been introduced to the region from Africa via the slave trade. So in 1941 was the first time that there was a serological report, a serological confirmation of dengue 2, but previously there were many written reports of dengue-like disease. Between 1941 and 1963, as I said, there was only dengue 2 up to that point. The reports of dengue-like illness were of mild disease. It was mainly dengue fever, and the outbreaks were restricted and self-limiting, were geographically restricted and self-limiting. Between 1963 and 1981, we had the introduction of dengue 1 and dengue 3. And during this time, we saw larger outbreaks and larger epidemics, and sporadic cases of severe disease were reported. In 1981, there was a big change. We had the arrival of both dengue 4 and dengue 2. By that time, the dengue 3 that was in circulation previously had disappeared. It was a genotype 4 dengue 3. And so we had dengue 2, 1, and 4 in the region. And when the dengue 4 and 2 arrived is when we started seeing dengue hemorrhagic cases in the Caribbean, and we had this dramatic increase in cases. Subsequently, in 1994, we had a new genotype of dengue 3 arriving, and that's when we ended up with all 4 serotypes circulating in the region. And what we have now is larger, more frequent epidemics with increasing numbers of severe cases. Chikungunya, so like dengue, is showing this sort of global expansion. So as I said before, the 1950s, it was restricted to Asia and Africa. It was first isolated in 1953. And in the 50s and 60s, there were large outbreaks on the Indian subcontinent, but then it disappeared in about 1970. 2005 was a key year. because that is when you had an urban epidemic in the Indian Ocean islands, and that then spread to the subcontinent. So this is the sort of genesis of the global expansion of the chikungunya virus. From 2006 onwards, there were imported cases reported in Europe, in the USA, and Caribbean islands, but none of those went on to result in sustained transmission, except in the case of an outbreak in Italy. And then in 2003 is when we had the outbreak in St. Martin, which has now spread to other regions. Right, the strain responsible for the Indian Ocean outbreak it belongs to the ECSA genotype, and it's most closely related to those viruses from East Africa. All right. So since the arrival of the virus in St. Martin, it has been spreading mainly southwards. And today we have, at last count, this was June the 2nd, was over 6,000 confirmed or suspected cases of chikungunya virus in the region. Interestingly, the strain of virus that is that is responsible for the outbreak that we're now seeing in the Caribbean is not the same one that was expanding out of the Indian Ocean. It belongs to the Asian subtype, as Asian genotype. Now, this type of viral emergence occurs regularly. Dengue and chikungunya are not unique in this. They're not unique in any way. And emergence of viruses can be a result of different factors. You can have evolutionary changes that affect things like host range, the virulence of the virus, the rate at which it's transmitted. You can have changes in host population susceptibility. For example, HIV-associated immunodeficiency has resulted in the re-emergence of TB. And things like reduced vaccine coverage as well can foster disease and virus emergence. And then you have the most important, which are the ecological changes that increase the probability of exposure of susceptible individuals or populations to infected reservoirs or vectors. And in most cases, this is what is driving emergence. And we mentioned them in the previous talk, things like urbanization. There's also the rapid global transport networks, etc. In the case of dengue itself, in terms of the global expansion, one of the very important factors was the movement, World War II, and the changes that occurred after that, movements of large populations, and also the expansion of the transport industry. And what continues to sustain it is the global population growth, urbanization, inadequate infrastructure, as we talked about, which facilitates breeding of mosquitoes and therefore increased dengue virus, dengue burden. Now, in the case of chikungunya, many of those factors are important. This vector is also the main vectors, Aedes aegypti and Aedes albopictus, would be influenced by the factors that I've showed before. But another important factor there was increased tourism in the Indian Ocean region, which resulted in the opportunity for expansion. There are also evolutionary changes in the virus that have been associated with its ability to replicate better in Aedes albopictus, so that was another important factor. And then, of course, the immune landscape is an important consideration. This is a virus which was restricted geographically to Asia and Africa, and is moving out into areas that have never met it before. So that's you have an introduction of the virus into naive populations. So what happens after invasion? Once the virus arrives in a new region, how does the epidemic, what's its epidemic behavior? And this is one of the things that we've been interested in our research. What sort of factors underlie epidemic behavior following introduction? What determines the rates and directions of viral spread? And what are in particular for us, what are the peculiarities of viral gene flow, viral movement in a mainland island such as ours? And we feel that identification of those factors underlying pathogen spatiotemporal dynamics and their relative contribution will allow us to better model disease spread and facilitate better surveillance and control. So some of the factors that could affect a mosquito-borne virus is like a mosquito-borne virus such as dengue or such as chikungunya would be the rates and directions of human movements, things like the geographic distance would influence that, the level of connectivity between areas, historical and socioeconomic links. So, Jamaica is closer to Puerto Rico than it is to Trinidad, but there's probably more a chance of a virus moving from Jamaica to Trinidad than from Jamaica to Puerto Rico if it's going to be carried by humans, infected humans, because of our links. Then, of course, there's the mosquitoes, rate and direction of mosquito movement, and also the availability of susceptible human hosts. So what's, things like human population density, etc., the immune landscape. So, and finally, availability of appropriate vectors is obviously an important consideration. Now, in order to understand those factors, you can use a traditional epidemiological approach. However, that requires extensive surveillance, and we heard from the previous talk that some of the types of data that we really need in order to be able to do this using a traditional approach are difficult to come by. Incidence rates, where are cases happening, how many cases are happening. And in our setting, often deficiencies in public health infrastructure can frustrate the collection of accurate and timely data. So it's difficult to get the type of data you need. For example, we looked at some samples from the Trinidad Public Health Laboratories during 2011. It was a 2-month period. And out of 710 samples that we selected from this 2-month period, 11.5% had clinical data. And of those, only, of those 82 that had clinical data, only 20 of them had both the date of onset of illness and information on symptomology. At the same time, we also looked at demographic data, and of the 710 samples, only 73.9% had any demographic data, and that was 525 samples, and of those, only 2 had the address, the age, and the sex of the individual. Now, this was during a period of, it was a large outbreak, so there might have been, the system might have been overburdened, but it's a difficult situation. So in addressing these questions, we would like to suggest, what we're trying to do is to use a different approach, a phylogenetic approach, where we rely on the history that the virus has encoded on its own genome. We sample viruses from the populations, we sequence their genomes, and we can create these data sets of virus sequences, which we can then exploit using a variety of computational tools. So we can, in addition to inferring things like the evolutionary relationships between sequences, we can also estimate the rates at which a virus populations involve in dates for which individual viruses or lineages first appeared. This type of thing you could probably get, if you had good epidemiological data, you could get it from that, but if you don't, you can sort of get out of the virus. You can reconstruct past population dynamics, you can reconstruct spatial dynamics, and you can create and compare models describing different patterns of geographic spread to determine which one best describes the patterns that you see. I'll skip past this. What I want to look at here is some of the work we've done with dengue 1 to 4 in the Americas. And you'll see that it's possible to recover information that is not available epidemiologically. So for example, if you look at dengue 1, the year when that was first reported in the Americas was 1979, but the genetic data suggests that it had been around for up to 5 years before. The country of first report was Jamaica, but the data suggests that it's very likely it existed in Grenada before that. Perhaps Jamaica had better surveillance than Grenada at that time. In the case of dengue 3, if we move down, we have the year of first report is 1992, but from the genetic data, the most recent common ancestor for those viruses existed since 1992. And we've correctly inferred Panama as the location of first report of where this virus existed, the first virus existed. And the country of first report in from epidemiological reports They are Nicaragua and Panama. So this is the type of thing that you're able to do with genetic data. And you can, okay, that has gone. There was supposed to be a video. Can you just skip to the next slide? What I was going to show was a short movie of the spread of the virus in the Caribbean, the pattern of spread that was inferred from genetic sequence data, not based on epidemiological data, but information that. Would you be able to start it for me? All right, let's skip to the next slide. We'll leave that out. All right, it seems to be working now. So this is a pattern of spread for dengue 1 that was inferred from genomic sequence data. We have virus isolates for which we know the date and the location from which they were isolated, and we can reconstruct the pattern of viral spread. Now, this was done for dengue 1, 2, 3, and 4. And you get a similar pattern of rapid dispersal followed by more localized maintenance. What is interesting to look at is to explore the factors that underlie the patterns that we've observed. So what is it? What predicts the rate and direction at which these viruses move? Some of the predictors that have been looked at are the distance between countries, the population size in the donor country, the population size in the recipient country, the distance between the closest and largest cities, the amount of air traffic between regions, and we've also looked at a number of markers or proxies for urbanizations, the percentage of the population living in an urban environment, human development indices, percentage of forest cover. And basically what we've pulled from these analyses is that there's more intense virus movement between nearby regions, more intense movement between countries with more air traffic between them. These seem a bit intuitive. But there's also more intense movement from smaller to larger populations, less intense viral movement out of countries with high urban populations, and less intense movement out of countries with high human development indices. So this whole pattern of spread in the Americas is best described by what is called a gravity model, where we see the dengue virus in a given location is more likely to move to a nearby and larger human population than it is likely to move to a smaller, more distant population. And so basically what you have is urban centers attracting the virus. We also, for dengue 1, 2, and 3, we also saw a good correlation with the amount of air traffic between regions. So air traffic seems to be a good predictor of spatial diffusion patterns. Just to end, I'd just like to present the people I need to acknowledge, the people who have collaborated with us on the dengue work specifically for the dengue. This looking at spatial temporal dynamics, and in particular, I'd like to highlight Orchid Alicock, my PhD student, who has done all the work inferring the patterns of dengue virus dispersal in the Americas and the factors underlying this. Thank you very much.